I would like to go into the presentation. Just give me a second. So, can you see my slides? Okay, so hello, good afternoon to everybody. First of all, I would like to thank to the entire organizing team for inviting me for this web-based lecture. Today, my presentation will be entirely based on the clinical laboratory approach in the diagnosis and monitoring of COVID-19. So, SARS-CoV-2 is, we know it's a beta coronavirus belonging to the family of coronavirus, which is so named because of the crown-like spike on the surface. There are 4 subgroups of the coronavirus known as alpha, beta, gamma, and delta. And some common coronaviruses like NL63 belonging to alpha coronavirus or HKU1 belonging to beta coronavirus are usually produce mild to moderate upper respiratory tract illness, for example, like common flu. Whereas this SARS-CoV-2 is a novel coronavirus that caused the disease that we call as coronavirus disease 2019 or COVID 2019. Well, we all know that SARS-CoV-2 caused an outbreak beginning from December 2019 in Wuhan city of China and has spread globally and consequently declared as pandemic by the World Health Organization. I would also like to briefly mention that the COVID-19 is the third coronavirus outbreak that we have observed from last 20 years after the human coronavirus such as MERS-CoV that caused Middle East respiratory syndrome and SARS-CoV that caused SARS. And they have caused more severe respiratory illness with higher rate of morbidity and mortality. SARS and the MERS altogether caused less than 2,000 deaths, while COVID-19 already affected over 7.4 million people with the global death of over 417,000. We are still quite far from over 50 million casualties that we have seen during the Spanish flu pandemic in 1918, but it has been calculated that COVID-19 may result similar casualty if appropriate preventive measures is not undertaken. If we see the structure of the SARS-CoV-2, it is an enveloped virus with positive sense single-stranded RNA genome containing 4 major structural protein known as S protein, which actually contain the receptor binding domain, envelope E protein, membrane protein, and the nucleocapsid protein N, along with additional genes such as ORF1A and B, ORF3A, ORF10, and so on, which encode the accessory proteins, including RNA-dependent RNA polymerase that is responsible for the viral replication. And in terms of the molecular size, we can see the biggest protein include ORF1A and B, followed by the spike protein. Since the virus can multiply in the cells of both upper and lower respiratory tract, the disease can spread from person to person through the droplet from the nose or mouth while person speak or breathe or cough or talk. Droplet can basically travel at the speed of 1 meter per second, traveling of 1 meter distance, while other activities like the breathing or coughing or sneezing can cause the droplet to transfer over 6 meters of distance. So as we can see in this simulation, that the how micro droplet generated during sneezing or simply talking can continue to remain floating in the air for hours and spread throughout the closed space. Therefore, the wearing of masks can decrease the production of the microdroplet, and also the opening windows in the closed space for a few minutes every hour can also decrease the risk of transmission considerably. Therefore, here in Japan, we followed strictly 3C rule, that is to avoid crowd space, avoid crowd places, and close contact setting. So if you overlap all these 3C in your daily life, the risk of getting infection can drastically increase. Regarding the pathophysiology, the virus uses spike protein to infect human cells using ACE2 receptors, and this ACE2 receptor are expressed in the cell surface of many tissues, including respiratory epithelia, nasal mucosa, or the pneumocytes, heart, kidney, gastrointestinal tract, and testes. The organ here I have shown in the red are more likely to get infected by virus and be potentially act as a high reservoir for this virus. And this table summarizes some biological source where virus can be detected with estimated rate of detection. Generally, the lower respiratory tract samples like bronchoalveolar lavas and sputum have higher rate of detection. Samples from upper respiratory tract like nasopharyngeal or oropharyngeal have generally over 70% of detection rates. And the signs and symptoms of are generally mild fever, shortness of breathing, cough, and sudden change in the taste and smell. But some cases may develop severe pneumonia. Generally, the data suggests that over 80% are asymptomatic or have a very mild symptom, while 18% can develop serious illness, and only around 2% are critically ill and are usually associated with other pre-existing conditions like old age, hypertension, and diabetes. Although the majority of the cases are asymptomatic, a recent study have shown that the viral load that we detected in the asymptomatic patients are similar to that of the symptomatic patients, which may suggest that the transmission potential of asymptomatic or minimally symptomatic patients. Therefore, the identification of the asymptomatic case are also equally important. It is now clear that the clinical laboratory play a critical role in the this global health crisis by contributing to patient screening or diagnosis, monitoring, and treatment, as well as by providing epidemiological surveillance. The laboratory investigation of COVID-19 include diagnostic tests like molecular testing, serological testing, and tests related to biochemical monitoring. Well, the nucleic acid amplification tests, or we call it as NAT, are currently the gold standard for the diagnosis of suspected case of COVID-19. The most common type of NAT that are being used is real-time reverse transcriptase PC polymerase chain reaction, which is used by both CDC developed assay as well as WHO endorsed assay. And also recently, the point of care testing based on rapid molecular tests are available in market, which are based on other technique like isothermal nucleic acid amplifications. While the serological test for COVID-19 is the analysis of the plasma or whole blood for the detection of antibodies, especially IgG, IgM, and IgA that are specific for SARS-CoV-2 antigens. including the spike glycoprotein and nucleocapsid protein. Testing methodologies in the serology vary from rapid diagnostic tests like use of disposable immunochromatographic lateral flow assay to ELISA or chemiluminescent immunoassay run by fully automated clinical laboratory instrument. And finally, also the routine biochemical, hematological, and immunochemical laboratory testing is essential in assessing disease severity, selecting appropriate therapeutic options, and monitoring treatment response. So first of all, let's talk a little bit about the nucleic acid amplification testing. The nucleic acid amplification testing is currently the gold standard for the etiological diagnosis of SARS-CoV-2 infection, which is mostly based on real-time reverse transcriptase polymerase chain reaction on the respiratory samples. There have been sorts of controversies on who to be tested, but it is clear that the test should be performed to those who really need the test, which will help not only to save the reagents, but also helps to prevent overwhelming clinical chemistry laboratory resources. So according to a WHO guide for global surveillance of COVID-19, the test should be performed in the suspected cases, and the WHO defined the suspected cases as one of the following. It can be a patient with acute respiratory illness and a history of travel or resident in the location reporting community level transmission of COVID-19 disease during the 14 days prior to the symptom onset, or a patient that with any acute respiratory illness and have been in contact with confirmed or probable COVID-19 cases in the last 14 days prior to the symptom onset, or a patient with severe acute respiratory illness, for example, having fever and at least one sign symptom of respiratory disease like cough or shortness of breathing and requiring hospitalization, and in the absence of alternative diagnosis that fully explain the clinical presentations. And WHO defined the confirmed case as the person with the laboratory, the person who positive for the laboratory test, irrespective of signs and symptoms. So there are different kind of specimen that can be used for NAT analysis. The sample can be both upper respiratory tract specimen as well as the lower respiratory tract specimens, for example, like sputum or bronchoalveolar levers. And also, although the virus can be detected in various biological fluids like blood, urine, and stool, these samples have actually very limited diagnostic applications or diagnostic value, but may be helpful to monitor the spread and the prognosis of the disease. The nasopharyngeal swab is considered as a gold standard sample for the detection of SARS-CoV-2 using molecular tests. It is very crucial that the adequate nasopharyngeal secretions should be collected to avoid the false negative results. And for the collection of nasopharyngeal swab, the swab is gently inserted into the nostrils to the nasopharynx, and the swab should be reached depth equal to the distance from the nostril to the outer opening of the ear, and we should leave the swab in the place for several seconds to absorb the secretion, and also it is recommended to rotate the swab several times before removing it in order to ensure the adequacy of the samples. So after collection, the swabs are placed immediately into a sterile transport tube containing either viral transport medium, VTM, or sterile saline, and the specimen can be stored at 2 to 8 degrees centigrade for up to 72 hours after collection. And if a delay in the testing or shipment is expected, then it should be stored at below 70 degrees centigrade in order to prevent the RNA degradation. So in order to various pre-analytical issues, including the inadequate procedure for the collection, handling, and transportation and storage of the specimen, the selection of the appropriate timing of the specimen collection is another crucial aspect to maintain the clinical sensitivity of nucleic acid testing. False negative results may be caused by the low viral load in the early and later stage of the disease. Therefore, it is very important that the sample is collected within the diagnostic testing windows. Generally, the study suggested that the peak of the viral seeding appears in 3 to 5 days after the infection. Therefore, it is the ideal or preferred time for the collection of the specimen. Furthermore, the timeline for the PCR positivity also different in the different type of specimen used. For example, the PCR positivity declines more slowly in the sample from the lower respiratory tract, for example, sputum, compared to the sample collected from upper respiratory tract like nasopharyngeal swabs. And the median period between the symptom onset and the negative PCR results has been reported to be 20 days, while ranging from 7 to 14 days. Therefore, by the end of third week, the viral shedding decrease drastically and may remain undetected afterward. Therefore, the timing in the testing is very, very important. The routine confirmation of the COVID-19 is based on the detection of unique sequence of viral RNA by nucleic acid amplification testing, such as real-time PCR, with the confirmation by, and sometimes it may need confirmation by nucleic acid sequencing. As we know that the coronavirus is an RNA virus, therefore it is necessary to convert RNA into its complementary DNA sequence using reverse transcriptase. Hence, the test is called as reverse transcriptase PCR or RT-PCR. The RNA is isolated from the samples and converted into its complementary DNA using reverse transcriptase enzyme, followed by its amplification by PCR into millions of copies for the detection. So the detection of the unique sequence of RNA fragment of the coronavirus is diagnostic, and this test can be used to diagnose COVID-19 in its early stage when the patients are highly infected. To perform this test, first viral RNA should be extracted. Several RNA extraction or purification kits are commercially available for a convenient, fast, and effective isolation. To extract the viral RNA, the sample is first mixed with the lysis buffer, which is highly denaturing in nature and usually consists of phenol and guanidine thiocyanate that degraded the viral protein core and released the RNA into the solution. The lysis buffer also consists of the RNase inhibitors to inhibit the degradation of viral RNA. And at this point, we can also add various internal controls like RP gene to ensure the quality of RNA extraction. Once the sample is lysed, the purification procedure is carried out by using a spin column. The lysed sample is transferred into the spin column and centrifuged. And this process is actually a solid phase extraction method in which the stationary phase is made up of silica gel, silica matrix, and in the optimal salts and pH condition, RNA molecule bind to the stationary phase while the protein and other contaminants are not retained and get eluted. After washing the column with the elution buffer, the RNA can be finally collected, which is free from protein and inhibitors and other other contaminated. And the concentration of the RNA can be measured by using UV spectroscopy to ensure the next step is the preparation of the reaction mixture for PCR amplification. In this step, the master mix is used, which is pre-mixed with the concentrate solution consisting of reverse transcriptase, nucleotide, reverse and forward primers, Tachman probe, and DNA polymerase. Finally, to complete this reaction, the RNA template is then added. Then the PCR mixture is loaded into the PCR plate and placed in the PCR machine, which is essentially a thermocycles. So the first step in RT-PCR is the reverse transcription. The DNA primer will specifically bind to RNA genome, and then the reverse transcriptase then is add DNA nucleotide at the 3' end of the primer, synthesizing new complementary of the viral RNA. Thus, the complementary DNA is synthesized, and the temperature and the duration of this step depend on primer and target RNA and reverse transcriptase. But generally, for COVID-19, we use the 50 degrees centigrade for 15 minutes for this process. The next step is the initial denaturation step for the denaturation of RNA-DNA hybrid. And it is generally carried out at 95 degrees centigrade for 2 minutes, and this process is also required for the activation of DNA polymerase and the simultaneous inactivation of reverse transcriptase. Well, the PCR, as we know, consists of series of thermal cycler, with each cycle consists of the 3 steps: denaturation, annealing, and extension steps. The denaturation step consists of heating the reaction chamber to 95 degrees centigrade for 3 seconds, and it is used to for the denaturation of the double-stranded DNA template. Next step, that is the annealing, the reaction temperature is decreased to 55 degrees centigrade, allowing the annealing of the forward primer to the complementary part of the single-stranded DNA template. In the extension step, the DNA polymerase then add the nucleotide and the complementary DNA strand is then synthesized. So after the first cycle, the double-stranded DNA thus obtained is after the first cycle, the double-stranded DNA target is then obtained. So then the denaturation of this double-stranded DNA is performed, yielding 2 single-stranded DNA molecules. Followed by the annealing step, where the temperature is again decreased, allowing the annealing of the reverse and forward primer to the complementary strand, along with the annealing of Tachman probe to the complementary part of the target DNA. If you talk about the Tachman probe, it consists of the fluorescent dye that is covalently linked to the 5' end of the nucleotide probe, and this fluorescent dye emit the fluorescence light when it is excited by the light source. Also, this probe consists of a quencher at 3' end, and the close proximity of the reporter to this quencher actually prevent the detection of its fluorescence. That means when they are close together, it does not emit any fluorescent signals. So in the extension step, then the DNA polymerase synthesize the new strand of DNA, and when the DNA polymerase reaches to the Tachman probe, the the endogenous 5' nuclease activity of the DNA polymerase clips this probe, separating the dye from the quencher, resulting in the production of the fluorescence signal. So as the Tagman probe is clipped, it makes fluorescence dye free so that it can generate the signal. So in the third cycle, again, the denaturation is a result, the conversion of double-stranded DNA into its complementary single strand, and then followed by the annealing of the primer and probes, and the extension to synthesize new strand of DNA by polymerase. So this cycle of denaturation, annealing, and extension goes on or can be repeated for over 45 cycles, and in each cycle, the number of DNA doubles, resulting in the synthesis of billions of copies of DNA. So in each cycle, the number of free fluorescent dye increases, thus the fluorescence signal is increased exponentially. So for the measurement of the fluorescence signal, the filtered light from the lamp is reflected off to the mirror and passes through the condensing lens, which is focused on the center of each well, and the fluorescent light emitted from the well reflected off the mirror passes through the emission filter and is detected by CCD camera. And each cycle of PCR, the light from the emitted fluorescent dye is detected by CCD camera, which convert it into digital data. Therefore, this method is known as real-time PCR because it allows the monitoring of the progress of PCR reaction as it occurs in real time. Sorry. The molecular assay used to diagnose SARS-CoV-2 are designed to detect specific target viral gene and regions, including S or spike, envelope, or nucleocapsid protein or gene, which represent the 3 or 4 major structural protein of the virus, as well as other gene that can be targeted includes the RNA-dependent RNA polymerase, RP gene, and the open reading frame 1A and B region. So various kind of the target gene has been applied in the diagnosis in the diagnosis of the COVID-19. So actually, many assays today are trying to target more than one gene to ensure the specificity of the test. So this slide shows some of the criteria used to assess whether the test is positive as recommended by WHO. We need to look at two different reasons. To consider a case as a positive in the area with no COVID-19 virus circulation, one of these two conditions need to be made. First, the positive NAT result for at least 2 different targets of COVID-19 virus genome, of which at least one target is preferably specific for COVID-19 virus, or one positive NAT result for the presence of the beta coronavirus and COVID-19 virus further identified by sequencing partial or whole genome of the virus. So whenever there is a discordant result happen, the patient should be resampled, and if appropriate, the sequencing of the virus from the original specimen or of that from amplicon generated from NAT should be obtained to provide the reliable test. Whereas in the areas where COVID-19 virus is widely spread, a simple approach can be adopted in which the screening of RT-PCR of a single discriminatory gene is considered to be sufficient. So, molecular test for the gene this slide shows the various potential pre-analytical and analytical factors that can affect the PCR testing. So one or more negative results do not rule out the possibility of COVID-19 infection, as there are number of pre-analytical and analytical factors that can lead to negative result in an infected individuals. Most importantly, it includes poor quality specimen that were collected very late or in the early stage of the infection. This specimen that were not handled or shipped appropriately, and also the test can be affected by various analytical related factors like virus mutation or PCR inhibition that can also lead to a false negative result. Therefore, if a negative result is obtained from a patient with a high index of suspicion for COVID-19 infection, particularly when only upper respiratory tract specimen were collected, additional specimen, including those from lower respiratory tract, should be collected and tested. So, also, another issue is about false positive result. For example, several countries have reported the cases in which patients who believed to recover from the COVID-19 being tested positive again for virus. Japan is the first country to report such cases in which the virus test was positive in the recovered patient. A few weeks earlier, South Korea reported over 260 clinically recovered patients with a negative virus test result later tested positive when retested after subsequent day. And the data from Wuhan, where the outbreak emerged, shows that about 5 to 10% of recovered COVID-19 patients tested positive for virus. Again, rising alarm that the virus might be able or capable of reinfecting the recovered patients. So we made some detailed investigation of such cases and reported that there are basically 4 reasons for the positive PCR results in the recovered patients. The first one can be the testing error. So the false negative results are not uncommon in PCR testing. If the PCR test that is performed before discharging a patient is false negative because of the flaws in the testing process, or PCR flaws in the if there is a flaws in the testing process and the PCR result get positive during follow-up, the patient after recovery, it is not appropriate to say that the patient have been reinfected. Further, I already mentioned earlier that the timeline of PCR positivity is also different in the different type of specimen used. PCR positivity declines more slowly in the sample from lower respiratory tract like sputum compared to the upper respiratory tract sample. Therefore, it is possible to get negative PCR result from the nasopharyngeal swab and positive from the sputum during the recovery phase. See, next can be the reinfection, although there are no enough evidence to ensure that the patient once recovered from the virus cannot get reinfected. However, reinfection appeared to be extremely unlikely. To this date, no human reinfection of this novel coronavirus disease has been confirmed, and there is no evidence that the patient who tested positive for the virus after recovery can transmit the disease to other person. A recent article in the Journal of American Medical Association suggested that the recovery of the COVID-19 might confer some amount of immunity against the reinfection, at least temporarily. The third reason can be the reactivation of the virus. In the most cases, our immune system is able to destroy the virus. However, in some people, virus remain inactive for a certain period of time, and remain hidden from our immune system. However, this inactive virus may get reactivated again when the person's immune system is weakened, and so further studies are actually needed to explore the possibilities whether this COVID-19 also act by reactivation. The most important cause for the false positive reaction is actually the detection of dead viral fragments. As in the recovery phase of the disease, what happened is the virus is killed by our immune system, but the dead viral particle may reach the dead cells containing dead viral particle are exfoliated and coughed up into the. Since the dead viral fragment of the virus, it can be detected by PCR, resulting in false positive results. Furthermore, a report from the Korean Center for Disease Control and Prevention states that they were unable to isolate the live virus from the sample of the individuals who have recovered, but again demonstrated positive PCR, indicating that the test in these recovered patients are more likely to be false positive. Therefore, it is very crucial to understand that PCR test is a test to detect RNA of the virus, but not the live virus itself. In other words, positive PCR reflects only the detection of viral RNA, and it does not necessarily indicate the presence of a viral virus itself. In that case, the virus culture should be performed in order to rule out the reinfection or reactivation. So, I'll shortly like to talk about the some commercial kits that are available now after the outbreak of COVID-19 started. We can see the hundreds of commercial molecular kit has been produced by the manufacturers around the globe. As of yesterday, a total of 78 commercial kits were approved by US FDA under emergency use of authorization, and many more are in pipeline. So there have been a few studies evaluating the comparison of the analytical and clinical performance of various molecular testing. This particular study evaluated the analytical and the clinical performance of four major SARS-CoV molecular diagnostic assay that includes the modified CDC assay, the assay from Simplexa, and the Eplex, and the Panther fusions. And they have analyzed the 51 positive and 53 negative nasopharyngeal swab specimen from the symptomatic patients, and the analytical and the clinical performance, including the positive percentage agreement and negative percentage agreement, were analyzed. And the results found that all the assay performed relatively well, with no significant difference in the analytical and clinical performance were noted. In the last few weeks, a number of publications are available assessing the clinical and analytical performance of widely used systems in the clinical laboratory, such as Roche-Cobas and Abbott system. And these studies shows that these systems for SARS-CoV-2 assay are acceptable for clinical use, and the use of fully automated system with a high throw output will be valuable in accelerating the pace of COVID-19 molecular testing in the coming days. Another aspect in the molecular testing is the point of care testing, or the rapid testing, which can actually revolutionize the COVID-19 testing. And these are the portable device and can easily identify various nucleic acid target within a few minutes to hours. Currently, certain systems have received US emergency use of authorization, the FDA authorization. And some of the popular POC test systems include the Export Express from Cepheid, and ID Now from Abbott, Aquila SARS from Mesa Diagnosis, and Spartan Cube from Spartan Bioscience. And this point of care testing are really interesting because they are technically very simple and can be set up in the very small scale laboratories that will help to diagnose the COVID-19 in the coming days. So particularly the export express system, if you see, this one is the first to receive the US FDA clearance, and the large number of the clinical laboratories have been using this system for the number of infectious disease diagnosis. And technically the procedure for this export express is very simple. We just need to apply a sample into the cartridge, and the cartridge is inserted into a machine or system, and the result can be available within 45 minutes. Another system, that is the ID Now system from Abbott, is another system that has actually drawn the attention in the laboratory testing of COVID-19. In this method, a test device is inserted into the test base holder, and the sample receiver is inserted into the sample receiver holder, followed by the mixing of the swab specimen for approximately 10 seconds. And after that, a test cartridge is used to transfer the sample receiver into the test base, and the result is available very rapidly. That positive result can be seen as early as 5 minutes. So the whole process is completed in 5 to 13 minutes. So this is really very exciting system that will certainly help in the prompt diagnosis of COVID-19 in coming days. In this slide, I have shown the comparison between Export Express and Abbott assay. The Export assay system is based on RT-PCR, and it is targeted on E and N2 while the Abbott ID is based on isothermal amplification and is targeted in RNase-dependent RNase polymerase gene. Abbott system appears to be superior with the analytical sensitivity, or the limit of detection of as low as 125 copies per ml, and the result is available within 5 to 13 minutes. And also in silico analysis showed no significant combined homologies with human genome and other coronavirus or human microflora that would predict potential false results. So next part, I would like to move to serological testing. The serology testing for COVID-19 can be defined as analysis of the plasma or the serum or the whole blood for the detection of antibodies, especially the IgG, IgM, or IgA that are specific for SARS-CoV-2 antigens, including the spike glycoprotein and nucleocapsid protein. It is used to assess the potential exposure of COVID-19, including those that may be asymptomatic or have recovered. The testing methodology vary from rapid diagnostic testing using the disposable immunochromatographic lateral flow assay to the ELISA and chemiluminescent assay, or it can be the neutralization assay that gives very valuable information about the presence or absence of antibodies that are actually able to inhibit the virus growth. Therefore, it indicates the protection against the future infection. And the neutralization assay is particularly important because the neutralizing antibodies from the convalescent plasma donor have been used in the treatment of COVID-19, and there are a few studies that have shown that the treatment with the convalescent plasma can actually lead to the clinical improvement. So when the coronavirus enter in our body, it stimulates the immune system to fight against the infection. And one way of the defense system is to produce the antibody that can destroy the virus, usually the patient start to produce the IgM type of antibody within 8 to 10 days of the infection, and will reach to the peak in around a month, and then a month of infection, and then it slowly declined. The IgA response developed early and coincide with the IgM peaking at around 18 to 21 days, and appear to be even stronger and more persistent than the IgM response. Patients also produce another type of antibody called IgG, which starts to rise in the blood within 10 to 12 days of infection and remains in the blood for a long time, at least for several months. So accumulating data suggests that the seroconversion occurs approximately around 7 to 14 days after the onset of the symptoms. At the present time, it is unknown how long these antibody persist in our body, and whether all these antibody produced are capable of neutralizing the neutralizing virus or not. At this time, we don't know the much information about the antibody. But what we can see is by the time when antibody appears, the viral shedding is already started decreasing. So that means the patients are already in the recovery phase. Therefore, the antibody testing have a very limited diagnostic value and should not be used to diagnose COVID-19 in the initial phase. So, despite the despite its limited use in the diagnosis, the serological testing can be helpful in confirming suspected cases, especially in the patient with milder or moderate illness or tested in the late phase of the COVID-19 that are not detected by the molecular test. Serological tests are also play important role to identify the past infections. It can be also used in various epidemiological surveillance and to determine the prevalence of the infection in the community. It is essential in the identification of the potential convalescent plasma donors and in monitoring immune response in the COVID-19 infections. It can be also useful to evaluate the effectiveness of the virus once it is developed. And the recent data suggests that the patient with severe COVID-19 have higher level of immunoglobulin G and immunoglobulin M titer compared to those with non-severe COVID-19. So the measurement of the antibody might also provide a very important or valuable information to assess the severity of the disease. However, there are also several drawbacks of antibody testing as reported by several groups. One of them is the false negative results. As we saw, as I mentioned earlier, that this test is not suitable for identifying individuals at the infectious state, and thus it have a very low positive predictive value. Moreover, the antibody production may be limited in the old age and the person with the immunocompromised conditions. Similarly, the false positive results are also of concern because of their potential cross-reactivity. The antibodies can cross-react with the antibody generated from other common cold-causing coronavirus because of their structural similarities. For instance, it can cause cross-reactivity with SARS-CoV-1 as both share almost 80% genetic identity with very similar protein. So the cross or the false positive result of the serology testing is one of the major challenge in the diagnostic field. So serological assay currently available in the diagnostic manufacturer varies significantly in the methodology, antibody target, and acceptable specimen type. As of today, more than 90 serological assay has been submitted for FDA clearance, but only 20 have received emergency use authorization so far, which include 8 assays that measure total antibody, 6 that measures IgM as well as IgG antibody, and 6 companies that only measure IgG immunoglobulins. So here I have presented a list of some of the assays that has received the US FDA approval, like Abbott, SARS immunoglobulin, BioRed, Diasorin, Orthoclinical Diagnosis, Roche, and so on. Basically, these assays largely differ in terms of the methodology and antibody target. However, the overall diagnostic performance of serological testing has not been systematically evaluated, and more data are required to support their clinical utility in the different settings. So there are a very few report that shows the comparison of various serological assay. This one from the Belgium that was published last week compared the 8 different antibody testing. The study results shows that these assays were somewhat agreeable to each others, particularly for the IgG subtype. The sensitivity of the detection of IgG antibodies at 14 to 25 days after the onset of the symptom is more than 92% for all these assays. As you can see here, that the average time for seropositivity for IgM was not shorter than IgG, and the study recommended to emphasize the measurement of IgG to avoid the false positive result that generally showed by IgM type. So next, I'll just briefly talk about the antigen testing. Considering the complexity, economic burden, and limited availability of molecular testing, the antigen testing in the respiratory samples can be game changer in the diagnosis of COVID-19. So far, only one assay has received FDA, that is SOPHIA SARS immunoassay, and it is based on the immunofluorescence sandwich assay that can quantitatively detect the nucleocapsid protein antigen from SARS-CoV-2 in various respiratory specimens. Although it is easy to perform with comparatively very low cost than the molecular testing, the study have shown that it have a major disadvantage in terms of having low sensitivity, especially when we compare to the molecular testing. So in the last section, I would like to discuss on the biochemical monitoring of severe COVID-19. The various routine biochemical, hematological, immunochemical laboratory testing is essential to assess the severity and selecting appropriate therapeutic options and to monitor the treatment response. And there are now several studies that has been published showing various changes in biochemical marker due to viral tissue damage or immunological reaction against the viral infection, or lack of oxygen saturation in the severe respiratory distress syndrome associated with viral infection. The severe COVID-19 is associated with the increase in inflammatory biomarkers and is characterized by massive pro-inflammatory response, or what we call as cytokine storm, that is believed to be linked with multiple organ damage and failure. This is particularly important as many pathophysiology of the severe COVID-19 are actually associated with cytokine storm syndrome. The severe COVID-19 lead to rapid activation of the innate immune cells, and this figure shows how the cellular and humoral immune system is activated in response to the SARS-CoV-2 infection that ultimately lead to the hyperinflammations. The level of many pro-inflammatory cytokines like interleukin-6, interleukin-8, or TNF-alpha, and also various kind of the chemokines are elevated in the severe COVID-19 cases, and the increased level of these cytokines are also correlated with the severity of the disease and are also significantly associated with mortality of this disease. The cytokines measurement is not commonly available in the clinical laboratory practice. Therefore, we usually use a surrogate biochemical marker of inflammations like C-reactive protein, ferritin, erythrocyte sedimentation rate, and all of these has been implicated in severe COVID-19 and can be useful in assessing the disease, the severity of the disease. Also, the studies have found that the higher level of procalcitonin in the severe COVID-19 suggesting of secondary bacterial infection in such critically ill patients. Similarly, the increase in the D-dimer, which is a coagulation marker, also has been reported, and it may be linked with thromboembolic event in COVID-19 patients, including pulmonary embolism and disseminated intravascular coagulation. And finally, the routine hematological examination shows leukocytosis, neutrophilia, lymphopenia, and thrombocytopenia. Next, the various biomarkers for evaluating the function of heart, the liver, and the kidney are also fluctuated in the severe COVID-19 patients. The accumulating clinical data suggests the prominent cardiovascular dysfunction occur in the COVID-19 patients. The significant number of the critically ill patient presented with the acute myocardial injury as determined by the increased level of the cardiac troponin level and are associated with increased mortality. Similarly, the other cardiac biomarkers like anti-proBNP, myoglobin, and CKMB are also found to be elevated. The possible mechanism of such cardiovascular complications are largely unknown, but we believe that it may be due to the viral myocarditis or the cytokine-derived cytokine-driven myocardial damage. Also, the recent evidence suggests that the liver dysfunction may also very common in the severe COVID-19 patients. Several large-scale hospital studies have reported elevation of various liver enzymes like AST, ALT, and gamma GT. And also the previous data from the SARS epidemic in 2002 reported that over 6% of the patients developed the acute renal impairment, and the mortality of the SARS patient with acute kidney injury was more than 90%. Surprisingly, in the current data shows that the prevalence of the acute coronary injury in COVID-19 is comparatively low, as indicated by the elevated level of creatinine and the blood urea nitrogen in the blood sample of the COVID-19 patients. So, in conclusion, the clinical laboratory plays a very essential role in this fight against this global health crisis by aiding the screening and diagnosis of the COVID-19, by monitoring and the treatment of the patients, and also by providing support for various epidemiological surveillance. The laboratory investigation in COVID-19 includes diagnostic tests like molecular testing, which is the gold standard for the diagnosis, the serological testing that can be used to identify the past infection or to assist the epidemiological studies, and the monitoring of immune response in the COVID-19 disease, and also provide the tests related to the biochemical monitoring, for example, the routine biochemical hematological investigation that give a very valuable information to assist the disease severity and monitoring the treatment process. So finally, I would like to recommend this guide information on COVID-19 related laboratory investigation in the that can be freely available in the homepage of the International Federation of Clinical Chemistry and Laboratory Medicine, and also I will like to recommend that you can access the various information about the laboratory investigation in general or related to COVID-19, and this platform is also available in the mobile-based app. So this kind of resources can also provide you various information about the laboratory investigation in the COVID-19. So finally, thank you for your patience, and thank you for this opportunity to share some of my thoughts through these presentations. Thank you.
Sir, thank you very much, sir, for your nice presentation. Let's for 2 minutes we will have a discussion session.
Okay. Okay.
I myself, I have one doubt actually.
Yes.